Hello students, today we are talking about oncogenes. Now before we understand what are oncogenes, we need to have a good understanding of these proto-oncogenes. Now proto-oncogenes are basically normal cellular genes and they have a variety of functions in the cells. So basically these proto-oncogenes are housekeeping genes. They have a lot of function they perform in the cells. Some of the roles these proto-oncogenes perform in the cell are signal transduction. Now signal transduction is basically cell communication, that is communication from one cell to the other, and for communication the cell requires a signal molecule, and this molecule then it basically relays the signal, which is referred to as signal transduction. So many of the proteins, many of the proto-oncogenes, they code for signal transduction proteins. They also have anti-apoptotic functions. Now apoptosis is basically cell death, programmed cell death. And these proto-oncogenes, they have anti-apoptotic, that is they prevent cell death. So they have anti-apoptotic functions. They code for proteins which are anti-apoptotic. They have role in they function as transcription factors like MYC. So many of the proto-oncogenes, they function as transcription factors like MYC, which now these MYC basically they transcribe genes which are involved in your cell growth and cell promotion of cell growth. So they also promote cell proliferation. So these are some of the functions of your proto-oncogenes, that is signal transduction, anti-apoptotic proteins, transcription factors, and they promote cell proliferation. Now what happens is proto-oncogenes, they get mutated and they get converted into oncogenes. Now proto-oncogenes are normal cellular genes and they undergo certain mutations known as gain of function mutations, which convert these proto-oncogenes into oncogenes. Now oncogenes are cancer causing genes. Now some of the mutations which can convert a proto-oncogene into oncogene are point mutations. Now there are point mutations. Point mutations is at a single loci. Now these point mutations can be there in the regulatory domain. So you have a regulatory domain, and regulatory domain basically controls the protein. It basically activates the protein, keeps the protein in the on state, and also keeps the protein in the off state. So it basically alters between on and off state. Now when there is a point mutation, especially in the regulatory domain, so these point mutations, they basically keep the protein continuously or constitutively active. So therefore the protein which was early performing some function, so it used to do that function and then get turned off, but now it is continuously active. So if it was performing cell proliferation, so it was basically toggling between on and off. So sometimes it was doing cell proliferation, sometimes it was not, but now it is a constitutively active protein. So these point mutations basically lead to more cell proliferation. The second which is B type of mutation would be a gene amplification. Now gene amplification is basically where the dosage of the gene increases. Dosage means earlier you had one copy of the gene, but now you had more than one copy, that is let's say 4 copies or 6 copies of the genes. So because the copies have increased, therefore the number of protein molecules is bound to increase, because the RNA will increase and therefore the protein molecule will also increase. So this is basically this increases the amount of DNA, and this increases the point mutation basically increases the amount of protein, that is a oncoprotein. This increases the point of oncogene, increases the amount of oncogene. Third is chromosomal translocation. Now chromosomal translocation can be of an overexpression. Now overexpression is when you have a translocated promoter, that is the promoter has come from some other part of some other chromosome. Now this translocated promoter, basically what it does is it increases the expression of the gene which was earlier. I mean earlier there was a normal promoter, so it had some expression, but now the gene is getting overexpressed, that is the expression of the gene has changed and it has increased basically. Now this overexpression, best example of this overexpression chromosomal translocation is BCR-ABL translocation, which is also known as Philadelphia chromosome. And the last is you can have chromosomal translocation like the formation of a chimeric protein. Now in this chimeric protein, again you have a protein which is basically hybrid protein. So this part of the chromosome has come from some other chromosome. I mean there is some translocation from some other part of the chromosome, and now this part is actually this is a chimeric protein, and this translocated gene fragment, it is basically replaced a regulatory fragment. So now this protein is going to be constitutively active, and this is a oncoprotein. So these two will lead to oncoproteins, and these will lead to increased expression, that is overexpression of the oncogene. So this is how, these are some of the mutations where a proto-oncogene is converted into an oncogene. So that's it. Thank you. Thank you for watching. Please like, subscribe and share, and write us back at genesisacademydaily@gmail.com. Thank you.